Welcome, everyone, to Prairie Restoration in a Changing Climate. A few quick reminders before we get started. Please use the Q&A box on the right of your screen for all questions. We'll be monitoring those to pass them along to Olivia. And anything that doesn't get answered during the talk today, Olivia has graciously agreed to log back onto Whova later and go through and be answering any remaining questions. For any other comments that you want to make, please use the chat function on the right for those so we can have a lively discussion during the presentation. And you'll be able to access all of this, including the video recording, through the agenda tab when the presentation ends. So today, our presenter is Olivia Chapman, a senior undergraduate student working towards a BA in sociology and anthropology at St. Olaf College. Her focus is environmental social science with a particular interest in environmental anthropology. Her research explores the interconnection of nature and culture, how each shapes the other, and why it matters. With that introduction out of the way, I'll pass things over to Olivia.
Awesome. Thank you so much, Becky. I am so excited to be here. Will go ahead and share my screen so we can get this presentation going. All right. Does everything look okay from your end, Becky? Okay.
Looks great.
Awesome. Okay. So hi, everyone. Like I said, I'm so excited to be here. I'm Olivia. Thank you, Becky, for that awesome introduction. Like Becky said, I am a senior right now in undergrad. I'm at St. Olaf in Southern Minnesota. And I am super excited for this opportunity to get to talk about my summer research project with you all. So I did this project on prairie restoration in a changing climate with my research partner, Cecilia Kibble, who could not be here today, but wanted me to tell you hi. So hi from Cecilia. And we worked under the supervision of Dr. Naomi Rushing, who works at Gustavus. So with that said, let's get into it. So I wanted to start with a bit of context because I think it's important to talk about research, situate research in terms of the context, why it's important, and why we care so deeply about the work that we're doing. So just briefly, not that this is necessarily a reminder that this group needs as the prairie enthusiasts, but tallgrass prairie is one of the most complex and diverse ecosystems on the planet. It is a habitat to so many creatures and so much life, and it's also one of the most endangered ecosystems on the planet. Minnesota is less than 2% of its original prairie left over, and we're losing it day by day, largely to climate change. So that is why we wanted to explore research prairie restoration within the context of climate change. So our research, what did we actually do? So this is Cecilia, this is my research partner out in our prairie. We'll talk about these little tents behind her here in a minute. But just to give an overview, this research, it had two parts. So we wanted to know, overall, we wanted to understand, one, how our prairie is changing in response to climate change, and two, what can we do to help restore prairies considering those changes. So those were our big sort of guiding questions for our research, which then translated into our more specific research questions. So for the first part of our research, right, for that first question of what's actually happening, right, like what is changing, we worked at a specific prairie, a strandness prairie. It is protected by the Nature Conservancy. So we had the awesome opportunity to work there this summer. And so we wanted to know, how has the flowering time, the phenology of plant species in strandness shifted over the last 50 years or so? So that was the first part of our research. And then the second question, we wanted to know, working with this specific study species, the partridge pea, which I'm sure you've all seen before. It's this beautiful yellow flower that grows, anywhere prairie is. We wanted to know if the addition of two specific microbes, so AMF, which stands for arbuscular mycorrhizal fungi, and rhizobia, which is a bacteria, does adding those to the soil of our partridge peas as they're starting to grow improve their fitness over time? Now, right? So under like current conditions, but also under projected future climatic conditions. So as the climate continues to change, we wanted to mimic those conditions and see what that would do to the impact of AMF and rhizobia on our partridge peas. So those are our big research questions. And now getting into what we actually did. So what did this actually look like? So, well, I will say it looked like many long days in the prairie, lots of prairie bathing, lots of early mornings, late nights, and hard work putting together these open top chambers, which I'll talk about in a minute, planting over 2,400 individual seeds, and just it was a lot. It was a lot. We played in the dirt, but it was really fun. So more specifically for this first part, for this first question, the how are things changing question. We collected, I mentioned we worked at Strandness, right? So we went out once every week for 10 weeks to collect phenology data. So data on what plant species are there, what flowering stage they're in. And we did that for our whole 10-week period. And the reason we worked at Strandness is because we actually are lucky to have about 50 years of phenology data already collected by a professor named Ellen Ordway out of the University of Minnesota. She started this project in 1975 and went out and periodically collected, the same phenology data that we're collecting, and kept all of those records. So we have just like stacks of her data sheets to be working off of and contributing to. So that was why we chose Strandness, why we continued working there. And yeah, that's that first question. And then the second part, the experiment part of our research, we were really lucky to have access to the Anguseth Rindy Wildlife Management Area. It's near Nerstrand, Minnesota. So it's a DNR run wildlife management area. It's about, I think, 3 acres or so. So that is where we did our field work, and our field work consisted of planting 16 plots of 30 subplots that looked like this. So this right here is what a subplot would look like, and each of the subplots had 5 seeds. So that math adds up to a lot of seeds. But so we went out and we planted our 5 seeds in our 30 subplots and our 16 plots. And then we covered half of our plots, so 8 of the 16 plots, with these open top chambers. And these open top chambers, this was how we mimicked future climate conditions. And so this was our solution for how to research, what those microbes will do to the plant growth under future climate conditions. So these open top chamber design came out of the University of Minnesota. A researcher named Rachel Payne developed this, and so we worked with her idea and put these open top chambers together. So I don't know, okay, right here and on the side is my research partner, Cece. So you can have some scale. But they're pretty big. They were maybe, I don't know, 5 to 6 feet tall. And they were just PVC pipe and wood beams and gardening plastic that we wrapped the structures in, and then we staked them to the ground so that they wouldn't blow away. We did have some trial and error with that one. Some heavy winds and some good old Minnesota weather put us through the ringer in terms of our open top chambers, but we got through and ended up with some sturdy structures and some new insight. So like I said, we had 16 plots. 8 of them were covered by the open top chambers. And what we did in terms of the treatment, right? So like I mentioned, we were working with AMF, fungus, and rhizobia, which is the bacteria. So on a quarter of the plots, we put just the AMF. We just mixed an AMF concentrate in some water and poured it over the plot, just watered the plants like we normally would, but with the AMF inoculant. The other quarter, or another quarter, sorry, we did just the rhizobia. And then third quarter, we did both the AMF and the rhizobia, that same process, just mixing it into water and pouring it over. And then the last quarter, we left as our control. So we just watered them with regular water. And that's how we did it. So we planted our plants at about 5 weeks into our research period. So we let them grow for the rest of the 5 weeks. And then at the end of the 5 weeks, we harvested our plants and took measurements, biomass measurements. We measured root length. We measured nodulation, which I'll talk about more later, and recorded all of those things. And that's how we got our data. So it was a really interesting process. And this was both Cecilia and I's first sort of experience with field work like this. So it was really interesting and fun and challenging and all the things to figure out how to conduct this kind of field work and deal with the challenges, right, that came up as we went through. There were lots of twists and turns and lots of mice, which ate a lot of our plants, but we got through. So there is an overview of what we did and now what we found. So for our first question, research is ongoing, right? So I said, we're working with Ellen's data and we're collecting data now. This is an ongoing project. My professor is wanting to keep it going for as long as possible to get as much data as possible. So we don't have anything, definitive, but from what we have, and especially from that 1975 to about 1998, so about a 20-year collection period, we found an overall trend towards earlier flowering. So essentially, as temperatures have gotten warmer, prairie plants have started to overall flower earlier. So that gives us some really good indication of how prairies are responding to changing climates, that they are responding, right? So there is something there. And that gives us some indication of what we can do to help help support restoration efforts in the future as the climate continues to change. So that was really exciting to find that those trends through that data. And we're excited to keep going on that project as well. For the second part of our research, the experiment part, overall, our AMF and our rhizobia that we added to the soil of our partridge peas, they improved partridge pea growth in both current and future climatic conditions. So in both the plots that were covered with the open top chambers and the plots that were left to ambient conditions, we saw an overall improvement to the partridge pea growth when we used AMF and rhizobia in combination. So separately, they were each helpful, right? So AMF on its own and rhizobia on its own, they both help the plant in terms of fitness, overall fitness. They were both helpful, but we found, and it was really exciting for us to find, that when we combined the two, when we combined the AMF and the rhizobia, we got it maximized benefits basically. So we chose these microbes because they have been shown in previous research to create a tripartite mutualistic relationship with legumes, which is what the partridge pea is, right, as a pea species. So our research supported those findings, and that was really exciting for us because that's kind of what we were, going in, not necessarily hoping, because hope in research is something, but that's what we were thinking. So it was exciting to find those results. So this graph that I've included here is how we thought a lot about our results, how we thought about fitness, and how we thought about growth. So Nodulation, so the root nodules, this is how you can see, basically, this is how you can see that the AMF and rhizobia are working. So when the AMF and the rhizobia work together, they fix nitrogen on the roots of the partridge pea, and they create like actual physical nodules on the roots. So you can like, after we harvested our plants and dried them out and cleaned them off, we could literally count the number of root nodules, and that was a proxy for how well, like for how much the AMF and the rhizobia were supporting the plant growth. The more root nodules, basically, the more the AMF and the rhizobia were helping that plant out. And overall, as you see, we can see on the graph, the more root nodules, the bigger the plants were. So there are lots of fitness measures that we looked at, but this total biomass gives a really good picture of just what's happening. So the more root nodules, which means the more that the AMF and rhizobia are working, the bigger the plants were overall, like by a lot. So that was, again, that was really exciting for us to find. And that's kind of what we were expecting. That's what we expected based on previous research. But yeah, it was cool to see it come out of our own study. I do want to comment, just because I think it's interesting and it's something that we talked about a lot. So this purple line here is the rhizobia only. So this was the quarter of the plots that were treated with just the rhizobia. And it actually, like, as you can see here, the more, oh, there you go. The more root nodules we found on the plants, actually the smaller the plants were. And so we looked at a lot of literature and found similar findings in other people's research. And essentially what we think is going on here, and what other researchers have found, is that when rhizobia is used individually, right? So just like on its own, it will nodulate really quickly at the beginning, which takes a lot of the plant's energy. And so that plant's energy is growing toward making the root nodules rather than creating more biomass in total. So for the first 5 weeks here, which was our measurement period, the plants were spending a lot of energy, a lot of effort making root nodules, so they weren't going to be as large overall. But what we found, or what we not found, but what researchers have found, and what we believe would be the case with our research, is that after this period, there is a pretty sharp increase in plant biomass. So the energy that was spent creating the nodules has a negative impact at first on biomass, but then eventually, after the nodules are set and the rhizobia has had its time to work, the plant will shoot up, and it's, in the end, it's beneficial to the plant. So that was another interesting thing that we found and we talked a lot about. So that was an overview. That was a quick and dirty overview of our research. But I want to spend the last 3-4 minutes here talking about this idea of finding our place. So speaking to the idea of like how, we did this research and we spent so much time in this prairie, and how did that help us like find our place as researchers, as people, as stewards of the prairie. And it just, this is something that actually Cecilia and I talked about throughout the research process. So all this summer, this kind of idea is something that we were talking about a lot. We sat down, before this presentation and went over some things, and we had some similar ideas. Like overall, doing this research was an incredible experience. It, like, we learned how to do research, right, which is a huge thing, but we also got to be really familiar with this prairie, with our prairie site, our little half-acre plot. became kind of home for the summer. And it was really incredible to have such a like intimate relationship with this prairie, with this place, and to be working in it and to be working with it and to get to know it so well. And you know, it kind of felt like we were friends with the plants, you know, we knew what was where, and we knew like there was this one of my favorite stories is this blackbird, this red-winged blackbird, mean birds, but they're beautiful. This one guy would come and join us every time we went out to do our research, and he would sit on our fence post and just watch us the whole time. And it felt like just our little, I don't know, good luck charm or prairie friend or something. But it was incredible. And so I think being able to do this research, being able to get to know this prairie so intimately, and also knowing that the work that we're doing is going to help prairies in the future, it was really exciting, and it really brought a sense of efficacy and joy. Like, this is something that we want to keep doing in the future, and this is something that is fulfilling. And yeah, it was just an incredible summer. So we had some exciting research findings, but we also just had some really great like personal findings, right? Some like finding our place moments, playing in the dirt and planting so many seeds and doing so much research. So thank you.
Thank you so much, Olivia, for presenting your research. And I think that, yeah, I mean, all of the attendees of our conference, all of the prairie enthusiasts can really identify with what you were saying about the multiple levels that it is satisfying and meaningful to be really engaging with the prairie. You're doing science, the internal work as well. So thank you so much for laying all of that out. We have had a barrage of questions come in, so we'll get through what we can today, and then hopefully you'll be able to follow up later. Um, but we had a number of questions about the earlier flowering that you were mentioning, and this wasn't necessarily the focus of your study, but do you have any information about how that lines up with the behavior of pollinators? Are they also being active earlier, or is this a real cause for concern?
Yeah, that's a good question. And I definitely like don't have the authority to say anything definitive. But that is something that we talked about. And we're looking at, so as the shift isn't going to be a uniform one, right? So we're seeing that the plants are flowering earlier, but that doesn't mean that the pollinators are automatically, you know, sort of going to catch on. So there is going to be a gap, and that gap is going to be rough. And I don't know how long it would be, and what exactly would come out of it, but that is something that is being talked about in terms of like restoration science, is like, how do we contend with this like, I can't remember the exact term, but there's a term for it, and it's a good one. I can't remember. But how do we contend with this inevitable sort of gap in pollination? So yeah, good question.
Mm-hmm. Great, thank you. Yeah, and maybe if we can look up the term, we'll drop it in the chat. But okay, well, and so we also had some questions come in about the open top enclosures you were using. Could you just speak a little more to the specific ways in which they're mimicking climate change conditions? And this might be too speculative, but the kind of like scale, is it emulating what you're predicting 50 years in the future would be like, or 5 years?
Yeah. So these, they're basically mini greenhouses. So we our goal initially was to construct these chambers to sort of heat up our half of our plots. So that we were working on the model of like climate warming. And scale wise, that's a good question. I don't know if we had an exact scale. We were thinking a few decades into the future. So more of the like future.
Mhm.
But yeah, so that was our goal. Our goal was to heat up our plots using these open top chambers, because Rachel Payne, who designed these chambers, found that the hexagonal design with the this specific type of plastic, sort of concentrated sunlight, right? And kept things heated up just a little bit, while also still allowing pollinators and natural conditions, rain, wind, all of that to get to the plants. That's why it's the open top part.
Mhm.
Um, but we found, as fieldwork goes, that our plan did not go to plan. And we actually ended up finding that the open top chambers, while they did heat up our plants during the day, they actually cooled them down at night. So rather than having a consistently warmed plot or consistently warmed plots, we had plots that were experiencing extreme conditions, so the warmer and the cooler, which is another climate model that has been proposed in terms of how things will look in the next few decades. So yeah, we had to switch some things up a little bit there, but that is how our open top chambers worked. And you know, that's what ended up coming out of it.
Great. Well, and I think you've kind of already answered another question that came in while you were talking, but whether you guys were also looking at predicted increases in drought periods, but it
Mhm.
sounds like you were letting the rain fall and that wasn't one of the control variables in your experiment.
Yep, exactly. We talked about it, and actually, that is some research that we did last semester in the greenhouse. So it wasn't field-based research, but it was an element of what we did in the lab. And it was fascinating. We found, I mean, what you would expect to find, like drought conditions aren't great for plant growth, things like that. But yeah, no, the good, that was not one of our variables here, but it is a really interesting and important one.
Hmm. Well, great. We've got time for one, maybe two more questions. So one of them that came in, it was about the AMF and rhizobia that you were using in like pre-European colonization. Would there have been a natural or organic source that you were trying to replicate and restore, or is this the introduction of a new practice or something to help with prairie resiliency?
Yeah, that is such a good question. I'm glad you asked because one of the reasons that we picked AMF and rhizobia specifically is because, I mean, not only because of the prior research that's been done on them, but also because they are naturally occurring in this soil. So, like North American prairie soil. So there was already AMF and rhizobia in the soil that we were working with, but we wanted to know if adding higher concentrations of this naturally occurring resource would help support growth even more. And it did. And so our results, you know, were exciting because our research question was answered, but also because this is a really practical, cheap too. It was really cheap to get the inoculum because it is so prolific.
Mhm.
AMF and rhizobia are everywhere. So it was exciting because this is a practical and cheap and easily implementable solution to help sort of support prairie restoration efforts anywhere.
Well, lovely. I think I'm going to have time to sneak in one more question because you
Yeah.
already sort of started answering it. But someone asked about the practical applications for AMF and rhizobia on prairies that like individuals might be establishing on just a couple of acres. But it sounds like what you're saying is like this is totally scalable. It's going to help no matter how you can be doing it.
Pretty much, exactly. Yeah. And if like you personally are interested, we used Prairie Moon. They're awesome. Really great site.
Mhm.
And just ordered like a ton of AMF and a ton of rhizobia, and you just there are recipes online for how much to mix into water. And yeah, you just water plants like normal, and it's great.
Well, fantastic. Thank you so much, Olivia. There were more questions that came in that we didn't have a chance to get to, but we're at time. And so just I want to say thank you again for coming, taking the time to share this exciting research and your unique perspective on these things. It was wonderful to hear, and it's been really fun for me as well to see the active question and debate happening on Huvo while you're talking.
I love it.
Um,
Nice.
but yeah, thank you so much. And so for all of our attendees, we have a half an hour break coming up, and our next set of talks start at 2. I hope you're able to join us. Thank you very much.
Thank you.